So, very good evening to all in this auspicious evening of the Republic Day of India. We have been traversing through the 18th day of the ongoing lecture series of undergraduate studies in zoology organized by the Zoological Association of Burdwan. This evening, we have with us Professor Sudha Anjela Dhan, Sudha Anjela Dhan, Assistant Professor, Rabindra Mahavidyalaya, Champadanga, Hooghly, to deliver today's stipulated topic. Now, I have the honor to request Dr. Saroj Kumar Ghosh, Assistant Professor and Head, Department of Zoology, BN Mahavidyalaya, as well as an active member of the ZAB Seminar Committee, to formally introduce Professor Sudha Anjela Dhan.
Thank you, sir, for giving me this opportunity. It is my cosmic happiness to introduce the honorable speaker with everyone present here. Professor Sudha Anjela Dhan is presently working as an assistant professor in zoology of Rabindra Mahavidyalaya, Champadanga. She did her master's in zoology with specialization in entomology from Benares Hindu University in the year 2015. Professor Dhan has accomplished CSR UGC NET in the year 2016. She has more than 5 years of teaching experience. With these few words, now I am joyfully inviting Professor Dhan to deliver a lecture on Platyhelminthes for favor of undergraduate students of zoology. Over to you, madam, please.
Thank you, Saroj sir, and thank you, Manobesh sir, for giving me this opportunity to deliver this lecture. Uh, am I audible?
Yes, you are audible.
Okay, I shall share my screen.
Just check the sharing, okay?
Yes, yes, sir.
Okay.
Okay, is it visible?
Perfectly visible.
Okay,
Go through full screen.
yeah, okay. Is it visible now?
Yeah, it is visible.
So, the topic of my lecture today is Platyhelminthes. To all the students, I would like to say this is an important topic of your paper, that is CC1. And today's topic is, as you know, is Platyhelminthes, and we'll be discussing the different classes. We'll be classifying the phylum Platyhelminthes, and also we'll be discussing about Fasciola hepatica and Toenia sollum. So let's begin. So phylum Platyhelminthes, generally we know that this phylum consists of the flatworms. Normally we can say that this is a phylum which has flatworms. And as you can see here, that phylum Platyhelminthes consists of several flatworms, not only flatworms, but apart from flatworms, it also consists of flukes, tapeworms, and some monochenians. Tapeworms is very popular among us. We all know about tapeworms and also about flukes, but we also will come to know about flatworms and monochenians. Before going to Platyhelminthes, I would like to tell you about all the other phylum that we have studied earlier. Maybe you might be knowing about some of the phylum, and that is the phylum that have been enlisted over here. You can see that is Protozoa, Porifera, Cnidaria, Ctenophora, Nemathelminthes, Mollusca, Annelida, Arthropoda, Onychophora, Echinodermata. So today mainly we will be discussing about Platyhelminthes. Okay. So what is Platyhelminthes? The Platyhelminthes, this word comes from two Greek words. Okay, the first part of the word is platy and the second part of the word is helminthes. Platy means flat, okay, and helminth means worms. So platy and helminthes both together forms the word flatworms. Because generally flatworms come under Platyhelminthes, so the word is kept in this way. So let us discuss about some of the important characteristics of the phylum Platyhelminthes. First of all, the body is bilaterally symmetrical, as you can see, and it's dorsoventrally flattened. Okay, so in flatworms, we will see that important characteristic is the dorsoventrally flattened body. Okay, so top to bottom, if you press, then you can find a leaf-like structure, that is the dorsoventrally flattened structure. Okay, second is they are triploblastic animals. Okay, third, they are mainly soft bodied with protective mucus secretions. Okay, they can have certain protective mucus secretions to protect their body. And we can also see that the circulatory and the respiratory organs are absent in it. Okay, because directly the gaseous exchange occurs by diffusion. Okay. Next point is you can also find some anterior eye spots and also some chemoreceptive organs present in the platyhelminthes. Okay, then is they are generally hermaphrodite. That means both the male and female reproductive organs may be present in one individual. The next is the development is generally direct.
Sorry for disturbing, madam. Madam, kindly
Yes.
change the slide. Madam, kindly change the slide.
Yes.
Madam, kindly change your slide, please.
Okay. Okay. It's not changing. Okay. Okay.
No,
I'm
no, no,
sorry.
no.
I didn't realize that. Okay. 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 I'll just stop sharing and then again I'll present. Okay.
Yes, yes, yes.
Okay. Is it visible now? Is it okay now? I'm
Yes,
trying to change
yes,
the
yes,
slide.
yes, ma'am. Yes, yes,
Is
yes,
it
yes.
okay now? Okay. Okay. I didn't realize that. Sorry for the inconvenience. Okay. Okay, so I'll just tell you once again. Okay, so phylum Platyhelminthes basically it includes the flatworms, monogeneans, flukes, and tapeworms, as I already said, and these are some of the names of the other phylum. Okay, under non-chordates. So the platyhelminthes, this word comes from two words, as I said, that is platy and helminth. Platy means flat and helminth means worms. Okay. So some of the important characteristics that we were discussing is body is bilaterally symmetrical and dorsoventrally flattened. They are triploblastic animals. They are soft-bodied, but they also have some protective mucus secretions. And the circulatory and respiratory organs are absent because the gaseous exchange it occurs by diffusion. Okay, there are some eye spots, anterior eye spots, and some chemoreceptive organs also may be found in them. They are generally hermaphrodite, that means the male and the female reproductive organs are present in one individual. The development is generally direct, but in some, there is a free swimming or Muller's or Gote's larva, and in some parasitic forms, there are several larval stages. Like in Fasciola, if we can see Fasciola hepatica, we will see there are numerous larval stages. Okay, we will learn about them later. That is Miracidium, Sporocyst, Radia. Okay, Cercaria larva. There are certain larval stages. Okay, but in some we can see there are not much of larval stages. Also, they do have simple development also in the process. Okay. So last point is they occurs in all major habitats. That is, they might occur in aquatic mode also, terrestrial mode also, including the tissues of other animals, like in certain classes like Trematoda, Cystoidae. We can find there are some platyhelminthes, those are endoparasitic. Okay, so it can actually uh, dwell in the tissues of other animals. So when we classify this phylum, we can classify this phylum into four classes. First is Turbellaria, second is Trematoda, third is Monogenia, and fourth is Cystoidae. Turbellaria, it comprises of planarians, okay. Second is Trematoda, it consists of flukes. Third is monogenians, they are generally ectoparasites of some aquatic vertebrates, and cystoidae, they consist of tapeworms. Okay, so let us first discuss about Turbellaria. So the class Turbellaria has some important characteristic which I have mentioned over here. As you can see that they are mainly free living and aquatic. And Turbellaria, this name actually comes because these Turbellarians, some of the Turbellarians, they have some cilia which can actually cause turbulence in water. So that's why the name has been kept like this, Turbellaria. Okay, so some of the important characteristics are there. They are mainly free living and aquatic. Okay, so body size ranges from few millimeter to 50 centimeter. Okay, few millimeters to 50 centimeter. The length can vary accordingly. Then most turbellarians, they move by cilia, which provide the propulsive force and larger forms are flat and move by combined ciliary and muscular movement. So we can see in turbellarians there is normally the cilia actually with the ciliary movement it provides the propulsive force to move forward. Okay, so we can find this kind of activity in them. So the next is mucus produced from the gland cells lubricates and protects the body surface and is also used in the capture of food by entangling the prey. So as we discussed earlier, there are also some mucus secretions which help in certain way. The next point is the digestion is initially extracellular and then intracellular. Okay, so both forms we can actually find. Initially, it will be extracellular, but later on we can see that the digestion becomes intracellular. The next point is excretion by protonephridia. Okay, so protonephridia we will find over here, and protonephridia, as we all know, it is concerned with the excretion and also osmoregulation. Okay, here are two examples of turbellarians. First is planaria and second is bipalium. Okay. Second class is class Trematoda. As I said, Trematoda, it includes flukes mainly. Okay. So Trematoda, they are all parasites occurring especially in vertebrates. They are very good parasites, I can say. Okay. So these are, the shape is cylindrical or leaf shaped. Okay. If you see the structure, they will be leaf shaped. I will show you the diagram. Okay. So you can see example is Fasciola. You can see how the shape is. Okay. So next is there are suckers and acetabulum is present for attachment. If we see closely, we can find some suckers and acetabulum. Acetabulum is also a cup-like shape that is used for attachment. So these suckers and acetabulum mainly it helps the parasite to keep itself attached to the host. So you will find these structures in Trematoda. Okay. Next is it consists of subclasses like Digyenia and Aspidogastria. So these are two subclasses which you can remember. Okay, that is Digyenia and Aspidogastria. So subclass Digyenia, it consists of those endoparasites which is found in all classes of vertebrates and are commonly called flukes. Okay, so the word that we call liver fluke, that is actually the common name of Fasciola. Okay, so Digyenia, it comprises of those endoparasites which are flukes generally. Okay, and flukes are mainly the parasites of vertebrates. Okay, next is another subclass, that is subclass Aspidogastria. Okay, so Aspidogastrians are mostly endoparasites in the gut of fishes and reptiles. Okay, so mainly we can find Aspidogastrians as endoparasites in the gut of fishes. Okay, so these are the two subclasses, Digyenia and Aspidogastria, under class Trematoda. So examples are Fasciola and Schistosoma. Okay, so Fasciola, as you can see, this is the liver fluke we know. Next is the class Monogenia. So Monogenia, there is one important thing about Monogenia. Monogenia, as I said earlier, that Monogenia, it includes ectoparasites. Okay, it includes some ectoparasites of aquatic vertebrates. So these are mainly those kinds of parasites which do not have any intermediate host. Okay, so maybe that's why the name is Monogenia. Okay, because the ones who need two hosts in that to complete their life cycle, we generally call them digenetic. Okay, so Monogenia, it does not require any intermediate host to complete its life cycles. Okay, and they are mostly ectoparasites of aquatic vertebrates, particularly fishes. Okay, their body is dorsoventrally flattened and they have a large posterior attachment organ. Okay, they have a large posterior attachment organ that is known as the hapter, which bears hooks and suckers. Okay, so you will find this special characteristic in monogenians. Okay, next is the anterior end also have some adhesive glands. So you can understand these hooks, suckers, adhesive glands. They are actually why is it present? They actually help the parasite to attach itself to the host, because as they are parasite they need to attach themselves to the host to obtain nutrition from them, right? That is why these structures keep them attached to the host. Next is the life cycle is simple and having no intermediate host. Like I said before, in class Monogenians, we see that the life cycle is simple. Okay, unlike the intro Trematoda and Cystoideae, where there are numerous larval forms and also intermediate host, definitive host. Likewise, in this, we will not find any intermediate host. Okay. Example is polystoma. As you can see, you can see this part. Okay, this part you can see there are numerous suckers. Okay, numerous suckers, as I said before, they will have this structure, especially in monogeneans. Okay, second is dactylogyres. This is also another type of monogenean. Polystoma, as you can see, it is actually mainly seen in frogs. Okay, this is a parasite. This keeps itself attached to frogs. Okay, and the second one, that is dactylogyres, it keeps itself attached to the freshwater fishes, and it attaches itself to the gills of freshwater fishes. Okay, so these two are ectoparasites, okay, which come under class Monogenea. The next is class Cystoideae. Okay, class Cystoideae, it mainly includes the tapeworms. Okay, so these are endoparasites. Okay, the organs of attachment is in the form of hooks and suckers. Okay, so we will find hooks and suckers as well in class Cystoideae. And next is, we will not find any sense organs. Okay, there is absence of sense organs. Next is, each segment except the head and the neck is provided with one or two sets of complete sex organs. Okay, if we see the example of tapeworm, we will find proglottids. Okay, so proglottids, in this proglottids, what we will find? We will find the male and the female sex organs in it. Okay, and these proglottids, they actually detach themselves and come out with the feces, okay, from the host, okay, to infect another host. Okay, so we will see the process in the life cycle, but let me tell you that they have certain segments which are known as proglottids, and in it we will find complete sex organs. Okay, complete sets of sex organs. Okay, next point is, life cycle is complicated and involves a larval stage. So in here also we will find the different larval stages. Okay, so example is Taenia sollum, okay, which we call pork tapeworm. There is another one, that is Taenia saginata, that is the beef tapeworm. Okay, so Taenia sollum is the pork's tapeworm, as its intermediate host is pig. Okay, so we call it pork tapeworm. And second is, another example is the Echinococcus granulosus. Okay, these two are the examples of class Cystoideae. Okay, now there are two um, uh, topics that you have to learn about. That is, first is the life cycle pathogenetic control measures of Taenia sollum, and second is Fasciola hepatica. Okay, so we will very briefly discuss about this. Okay, I'll just tell you about its life cycle and then pathogenesis and the control measures of this parasite. Okay, so first take Taenia sollum. Okay, Taenia sollum, common name is pork tapeworm. Why do we call pork tapeworm? Because pork is actually the meat of pig. Okay, the meat of pig is called pork, and since this Taenia sollum, it also infects pig. Okay, pig is as like an intermediate host. Okay, so it infects pig, that is why we call it pork tapeworm. Okay, like I said, there is another one, Taenia saginata. It infects the cow or the cattle or the beef. Okay, that's why we call it beef tapeworm. Okay, and over here, as we see, it affects pig, so we call it pork tapeworm. So let us see what are the characteristics. First, it is usually creamish in color. Okay, normally we see in books that it's white, but it's not fully white. We can see there is a slight creamy color in it. Okay, so it is creamish in color. Its body is 1 to 5 meter long, and it is flattened like a tape. We all know how a measuring tape looks, right? So it looks exactly like a measuring tape. Okay, it's flat. Okay, it's tapered. Okay, so it looks like a tape. Then entire body is divisible into three distinct parts. Okay, if we divide its body, then we can divide its body into three parts. That is the scolex or head, the neck, and the segmentus strobila. Okay, left side you can see a diagram over here. Over here you try to see the topmost tip. Okay, that is the scolex, which is the head. And here we will find four suckers. Okay, here we will find four suckers. Like in the right side of the screen, you will see there is a picture over here. Okay, this is the scolex. Okay, the scolex, as you can see, there are four suckers, and on the top there are two rows of hooks. Okay, so why these are important? It helps this parasite to keep itself attached to the host. Okay, so it helps itself to keep attached to the intestinal mucosa of the host. Okay, that is why it is provided with hooks and suckers. Okay, so this scolex is also called the head. Second part is the neck. Okay, the neck region. It will have a small neck. Okay, Then is the segmented strobila, from where the strobulations will start. And as you can see, from the neck down, the size also increases. As it goes further downward, the proglottid size also increases. And we can also divide the proglottid into three parts, that is immature proglottid, mature proglottid, and gravid proglottid. Okay, immature proglottid, the sex organs are not well developed. Okay, mature proglottid, yes, the sex organs will be developed. And gravid proglottid, it will consist of eggs. Okay, there will be a mature ripe uterus full of eggs, which is actually because these gravid proglottid, they detach themselves, okay, which through a process called apolysis, and they come out with the feces from the host body to again infect another host. Okay, so we can see as you go further downwards, you will see the proglottids are of different types, that is the immature, then mature, and gravid proglottid. So it is the gravid proglottid that detaches itself first. Okay, so this is about the structure of tapeworm. Okay, next point is elongated body of tapeworm is divided into a great number of parts or segments called proglottid. As I said before, these all segments are called proglottid. You have to remember this. Okay, so these are known as proglottids. Okay, and as I said, this is the structure of the head where there are 4 suckers present and 2 rows of hooks are present. Okay, and these all help itself to get attached to the remain attached to the host's body. Okay, so this is about the structure. Next, let us also see some important points. Okay, so about Taenia soleum, it completes its life cycle in 2 hosts. Okay, it is digenetic. It needs 2 hosts to complete its full life cycle. Okay, so in case of Taenia soleum, it will be man and pig. Okay, so the adult dwells as internal parasite in the small intestine of man. Okay, so where will we find the adult? We will find the adult in the intestine of human being. Okay, and human being is the primary or the final host, where it is anchored to the intestinal mucosa by its scolex. Okay, as you all saw, the scolex has suckers and hooks. Okay, so with the help of those suckers and hooks, it will attach itself to the intestinal mucosa of the primary host, which is the human being. Okay, it has no mouth or digestive cavity but absorbs the host's digested food through its body wall. So over here we can also learn about some important characteristics of Taenia soleum, that it does not have any mouth or digestive cavity because already it is absorbing the host's digested food through its body wall. Okay. Next is the larval stage occurs in the tissues of a secondary or intermediate host, which is usually pig. This Taenia soleum has a larval stage, okay, which is occurring in the secondary host, which is also called the intermediate host. And here the intermediate host is usually the pig. Okay, and one more thing I'll tell you. You will study about primary host, okay, secondary host, and in some places there will be written definitive host or intermediate host. So who is the primary host? Primary host is the one where the sexual reproduction occurs, which is very important to continue its life cycle, right? So likewise, we can name primary host as the one in which the sexual reproduction will occur. Okay, so we can divide primary and secondary. Okay. Now, let us talk about its life cycle. So how does the life cycle start? So here you can see, if you see the adult tapeworm, okay, if you see the adult tapeworm, then you will find over here that it has proglottid, okay, and the last proglottid is called the gravid proglottid. Okay, so gravid proglottid, it is getting detached. Okay, it is getting detached and it is coming out from the primary host's body, which is man, and through feces it is coming out. Okay, that means in the infected man, okay, if a man is infected with this tapeworm, then in his feces the gravid proglottid will come out. And what does the gravid proglottid contain? It is full of eggs, right, to be released into the environment and to infect another person or another host. Okay, so it comes out, it is full of the eggs. Okay, and when these eggs gets contaminated, okay, with some other thing which is again consumed by the pig. Okay, so as you know, there are some pigs, or maybe those who farm pigs, okay, those who keep them in their farm, if they are not in proper hygienic condition, okay, if these pigs are consuming the human feces, then what will happen? These feces will actually go into the pig's stomach, okay, and then these eggs will reach the pig's stomach, okay. So the pigs get contaminated in this way. And now what will happen? For these eggs will, what happen? These eggs will actually turn into cysticercus. Okay, so this cysticercus will then get deposited into the muscles of the pig. Okay, these will get deposited into the muscle of the pig, and now we will call the meat of the pig as measly pork. Okay, so the meat that is already infected with the cysticercus, okay, we will call it measly pork. Okay, so the cysticercus, they develop in the pig muscles, and if this meat or if this measly pork is consumed by again another human person, okay, which is not infected, for if you suppose if it's not infected, then what will happen? If he doesn't cook it properly, or if the meat remains undercooked, okay, then this cysticercus will get transferred to the intestine of this another human being. Okay, so these humans will acquire the Taenia solium infection by ingesting the raw or the undercooked pig meat, okay, that is infected with the cysticerci. Okay, so like this the life cycle will keep on continuing. Then in the gut, this will again mature, okay, mature and become to adult tapeworm. Okay, this will again release its gravid proglottid through the feces into the open environment. It will again get consumed by pig, or sometimes maybe if we do not wash our raw vegetables, okay, properly and consume it anyhow, then also what happens, these contaminated raw vegetables also through consumption we can get this cysticercus inside us. Okay, so this is how the human beings also sometimes get infected with this cysticercus. Okay, and this cysticercus is very dangerous. Okay, and the disease that is caused due to cysticercus, we call it cysticercosis. Okay, and this happens when the cysticercus gets into the human tissues such as brain, eyes, muscles. Okay, then it becomes really very difficult, okay, to remove from these important and vital organs. Okay, surgery is the only way. Okay, so you have to remember this is very important that we wash our vegetables, okay, properly, so that we don't consume contaminated food. Okay, and it's best to keep it boiled. Okay, if we boil and consume, it is the best. Okay, to be free from all kinds of parasites. Okay, so this is how this whole life cycle continues. Okay, so this is about the life cycle of tapeworm. Now let us talk about pathogenesis. Okay, what happens? Man may fall a victim to adult tapeworm as well as its cysticerci. Effects are referred to as teniasis and cysticercosis respectively. Okay, so there are two words that you have to remember, that is teniasis and cysticercosis. So teniasis, when man becomes a victim of the adult tapeworm. Okay, then we call it teniasis, and when it becomes a victim of the cysticerci, it is called cysticercosis. Okay, so teniasis, what is teniasis? Its symptoms include pain in the abdominal pain. Symptoms include pain in the abdomen. Okay, then nausea, anemia, increased appetite, digestion, and above all, nervous disorders of the type occurring in epilepsy. So these are the various symptoms with which we can understand that a person is actually infected with tapeworm. Second is cysticercosis. Like I said, it is very dangerous than teniasis. What happens? The encystment of the bladder may take place in the host's voluntary muscles. Okay, so this cysticerci or cysticercus that I was talking about, this gets deposited into the muscles. Okay, and not only voluntary muscles, the cardiac muscles also, and even in more delicate vital organs like liver, eyes, and brain. So when the cysticercus reaches the liver, eyes, brain, it becomes very difficult to remove, okay, from these delicate tissues. Okay, so cysticercosis of the brain, it also results in several degenerative changes and also necrosis in the brain, and the patient shows epileptic behavior. So it becomes very difficult. Okay, and it's far more dangerous than teniasis. Okay, but still we have some remedy, we have some treatment for this. Okay, so what is the treatment? Infection of tapeworm can be tackled by several anti-helminth drugs such as praziquantel, albendazole. So these are two drugs which you can take when you are diagnosed with teniosis. Okay, then removal of scolex can be brought about by surgery. So what is the scolex? The scolex is the head region. Okay, sometimes what happens with drugs, you can actually treat or remove the rest of the part of the body, but the scolex remains attached to the intestinal mucosa. So to remove that, you have to do surgery. Okay, sometimes. Removal of cysticerci from delicate organs like brain, eyes, and liver is also extremely difficult. Okay. Now, what are some of the prevention or control measures? Okay, there are a few which I have mentioned over here. First is consumption of undercooked and measly pork should be avoided. So we have to avoid measly pork consumption. Okay, and undercooked also should be avoided. Second is feces of infected person should be properly disposed of and destroyed. So if a person is infected and we know that, so we have to avoid we have to properly dispose, okay, dispose of the feces or infected clothes and all, okay. Then prevent the pigs from having access to the fecal matter. As we all know, the intermediate host is pig. Okay, so pig is also important over here. So we have to prevent the pigs from having access to the fecal matter of the infected humans. Okay, or any other fecal matter, because from there only the life cycle will be continued. So we have to prevent that. We have to stop that. Okay, so in this case, we have to be careful and we have to take proper prevention and control measures. Okay. So this was all about the Taenia sollum, that is the pork tapeworm. Second is the Fasciola hepatica, which is also called the liver fluke. About this also I will be talking in brief. Okay, so first let us see what is the life cycle. Okay, before that let us know about some of the characteristics of the liver fluke. Okay, so what is liver fluke? It is an endoparasite. It is an endoparasite. Okay, it stays inside the body of the host. It is an endoparasite which completes its life cycle in two hosts. It is also digenetic. It also needs two hosts. Okay. So the adult Fasciola hepatica is known as sheep liver fluke because it occurs in the liver and bile passages of the sheep, which is the primary host and causes liver rot. So what happens over here? Over here, this Fasciola hepatica, which is, as the name is given, liver fluke, it actually attacks. Okay, it actually attacks the liver. Okay, it causes the liver to rot. Okay, but because when it infects, okay, heavy infection will damage the liver. Okay, and ultimately can cause death also, death of the sheep also. Okay. So Fasciola hepatica, it spends a part of its life cycle in an intermediate host, that is a freshwater gastropod called Limnia truncatula. Okay, so as I said, there are two hosts over here also. It is also digenetic. The primary host is the sheep and secondary host is the freshwater gastropod. Okay, freshwater gastropod. Its name is Limnia truncatula. Okay, so its body is soft, oval in shape and dorsoventrally flattened and leaf-like. So how is the structure of liver fluke? It is soft. Okay, it is oval in shape and it's dorsoventrally flattened like the leaf. Its length is about 1.8 to 3 centimeter long. Okay, there are two small suckers, anterior and ventral. Both are devoid of hooks and spines. Okay, they do not have hooks like the one we saw in Taenia sollum. They do not have hooks, but they do have the suckers. Okay, they do have two suckers. Okay. So this is the life cycle. In the life cycle, you will see a part of its life cycle is completed in the sheep and a part of its life cycle is completed in the snail. Okay, snail, as we said, it's a gastropod, it's a limnia. Okay, so here what happens? Here you can see different larval structures over here. Okay, it's a bit complex than Taenia sollum. It's a bit complex. Why? Because it includes more than 2 or 3 larvae. Okay, like if we start from here, okay, so this is the adult fasciola. Okay, the adult fasciola, it will come, if adult fasciola will infect the liver of the sheep. Okay, and with the feces of the sheep, it will come out. Okay, when the sheep will excrete with the feces, this adult fasciola eggs will come out. Okay, so when the eggs come out, they will hatch. Okay, and if an aquatic water body is near, okay, or if a water body is near to it, then it will help the eggs. Okay, will help the eggs to develop into miracidium larva. Okay, and this miracidium larva is free swimming. Okay, it can swim and go to places. So if it has access to any gastropod, any snail, and especially in this case we are talking about limnia, if it has access to this particular snail, that it will infect this snail. Okay, it will infect the snail, it will infect its body. Okay, it will enter into its body and it will change from miracidium larva to the sporocyst larva. Okay, so from miracidium larva, which was a free swimming larva, when it infects the body of the snail, it will turn into sporocyst larva, and from sporocyst larva it will again turn itself into radia larva. Okay, and then from radia larva it will turn itself into cercaria larva. It is all happening where? Inside the body of this gastropod, inside the body of this snail. Okay, so inside the body of the snail, it will reach up to cercaria larva. Now from cercaria larva, it will come out. Okay, so the mature cercaria, it makes its way through the host tissues and migrates to its pulmonary sac. Okay, and from there it will escape into the surrounding tissues. Okay, so this cercaria larva will then develop into metacercaria larva. And how it will develop into metacercaria larva? It will lose its tail. Like here you can see the encysted metacercaria larva, it does not have any tail. Okay, so the cercaria larva, it will develop into encysted metacercaria larva, and this metacercaria larva, you can see it will be found in the blades of leaves. Okay, because as you know, the sheep like grazing in the field. Okay, and it grazes on grasses, it consumes green grasses. So these metacercaria larva, it will be attached to the blades of the leaves, and when the sheep will graze on those leaves, then it will again consume this. Okay, consume this metacercaria larva, and this metacercaria larva will then further develop into the adult fasciola in the liver. Okay, and then this fasciola will again reproduce, again produce eggs, and these eggs will again be released through feces. Will if it will find any aquatic water body near to it, then it will then develop into miracidium larva again, and then from miracidium again sporocyst, again radia, again cercaria, again metacercaria, again if the sheep consumes it, it will again turn into adult fasciola in the liver. So this way the life cycle will be completed both in sheep and in snail. So over here the definitive host will be the sheep. Okay, and the intermediate host will be the snail. Okay, so it is a bit complex, but still you can remember the different names, and through this way I have tried to explain it to you in the best easiest way possible. Okay, so I hope you have understood it. Okay, so let us talk about the pathogenesis. Okay, so the infection in vertebrate host, that is the snail. Okay, invertebrate host is the snail, vertebrate host is the sheep. Okay, so invertebrate host is the snail. So the infection in the invertebrate host, that is the snail, it results in the partial or complete destruction of the affected site, which is preferably liver or gonad. So in snail also it affects the liver or the gonad. Okay, and in vertebrate host, the sheep, the bile ducts as well as the liver may be damaged due to infection. Okay, so heavy infection may upset the normal metabolism of the liver, and the disease that is caused is known as liver rot or fascioliasis. Okay, so in vertebrate host, the liver, if there is very heavy infection, then it will cause liver to get damaged, and which is also called liver rot or fascioliasis. Okay, but there is also some treatment and prevention and control measures for this also. So here is a drug called triclabendazole, okay, which is also called Fascinex liquid. It is considered as the most common drug due to its high efficacy against adult as well as juvenile flukes. So you can use this particular drug. Okay, if any animal is infected, you can give this drug to that animal or sheep. Okay, and also a new drug which is called the compound alpha. It is also similar to triclabendazole, and it is being tested now. Okay, but there are still ways that we can prevent this infection. Okay, and those ways are like killing heavy infected sheep. If you find a flock of sheep is heavily infected, then it is better to kill those. Okay, then destroying eggs and manure of infected sheep. Okay, so if any sheep is infected, the manure may be destroyed. Okay, we will not use, because further it will again infect other healthy flock of sheep. The next is killing and checking snail population. Here this is very important because snail is an important part of the life cycle. So if we try to avoid this or prevent this, we can prevent this parasite also. Okay, so how we can kill and check the snail population? Ducks can help us, because ducks generally feed on these types of snails. Okay, so if we introduce ducks, then they can help to control the snail population. Then last is man can also avoid infection by consuming thoroughly washed and adequately cooked vegetables. So if we cook our vegetables properly, if we boil our vegetables properly, then we can avoid infection also. Okay, so this is the prevention and control measures, and I hope you have understood, and with this I come to the end of my lecture. Thank you very much. And if you have any questions related
So,
to this, you
yeah, yeah. So, heartfelt congratulations, Professor Shudha, for a nice and illuminating lecture. Now,
thank you,
the
sir.
session is open for interaction. Students, if you have any question, you may ask to the speaker. I think the speaker's email ID and phone number, etc., is given in the chat box, so you can collect it from there. But now you can interact directly with the speaker. Is there any question?
There is a question by Kishore Ghosh. What is the best molecular marker to carry out the phylogeny of flatworms? I'm sorry I cannot answer this because I have not done the study on this recently. If you have any questions related to the life cycle, maybe you can ask.
So, any more question? This is a part of molecular biology. This is not a part of bionomics. So, you can ask the question from the part of bionomics. Is there any question from part of bionomics or life cycle or pathogenicity, etc., etc.? You may ask the speaker. So, I think there is no question in this session. So it is time to give the formal vote of thanks, and I don't want to waste the opportunity to invite Dr. Shamdas Bandyopadhyay, Assistant Professor, Department of Zoology, Burdwan Raj College, as well as active member of the ZAB Seminar Subcommittee, to perform the job. Dr. Bandyopadhyay, please.
Thank you, sir. In continuation of the effort by the Zoological Association of Burdwan to let the students cope up with the unprecedented situation of the ongoing pandemic, arranging a series of online lectures is a great effort aimed at helping the students. On this auspicious Republic Day, I sincerely thank the organizers for giving me the opportunity to propose vote of thanks and acknowledge the contributions of the speaker. I extend vote of thanks to our respected speaker, Sudha Anjela Dhan, who is currently serving as an assistant professor in zoology, Rabindra Mahavidyalaya, Champadanga. We had an excellent opportunity to listen to the lectures and gain knowledge on various aspects of the phylum Platyhelminthes and its importance in the living kingdom with the help of the scheme of its various scheme of the classification and various examples and their economic importance. Thank you for letting the light of the knowledge ignite the inquisitive minds of many of our students. My gratitude to the respected speakers for teaching our beloved students. Also thanks to two of our active organizers, Dr. Ghosh and Dr. Mujumdar. Over to Dr. Mujumdar.
So with this, we are actually concluding formally today's session. Thank you. Thanks all.
Thank you, everyone.